Week we're going to talk about biodiversity and its influence on ecosystem processes. If you remember from the beginning of class, we talked about that there are different state factors that influence ecosystems and ecosystem processes or ecosystem function. And one of those state factors is organisms, which is what we'll focus on today. All right. So a big question in ecosystem ecology is understanding the influence of biological diversity on ecosystem processes. Now, when we use the term biodiversity, that can mean many different things. We could be considering the genetic diversity within a population. We can look at species diversity, which is the number of individual species and their relative abundance within an ecosystem. We can also look at functional group of species. So these are species that have some type of function or role within the ecosystem, and we'll talk more about what those functions might look like. We can also talk about the diversity of multiple ecosystems on a landscape. So a key kind of component is thinking about the identity of those species versus the richness or the number of those species, or key functional traits versus the diversity, species diversity. So why should we care about this? Well, it's important to understand the state factors control on ecosystem variation in both space and time. When we better understand how ecosystems operate, we then can start to begin to understand how humans can impact those ecosystems, and then how might we as humans, change our behavior or our management practices to have a positive impact on those ecosystems. So ways that humans can impact ecosystems is through habitat destruction, which could lead to an extinction of some species. Another way is through simplifying the ecosystem or the land. Maybe that's converting a forest system to a crop monoculture that's only one or two plant species. Um, the other is to introduce organisms that are not normally found within that ecosystem. And those can cause biological invasions if we have invasive species. So the variation in key traits can cause species to differ in their effects on ecosystem processes. Um, they could affect the biogeochemical cycle, so thinking about cycling of carbon, nitrogen, phosphorus, water. They can also have an effect on biophysical processes, such as like decomposition of nutrients or litter or the environment or the microenvironment. They can also have an influence on the trophic structure. And then they can have an influence on disturbance regime. If we get a shift in the dominant plant species within a system, we might shift from a plant species that is not adapted to a fire to one that is adapted. A good example of that is cheatgrass, an invasive species that's found across the western United States. It tends to burn more readily than the other plants that it outcompetes for outcompetes for. So a big question is what are diversity per se effects versus the functional trait effects? And here's just a graph showing some hypothetical relationships between species number on the x-axis and then the ecosystem process on the y-axis, which we could replace with a specific ecosystem process, say something like nutrient cycling or nitrogen mineralization. So there could be one scenario where as you increase the number of species, you can increase the species that are unique to the ecosystem, that you could have a linear relationship. So as you increase the number of species that are unique, you could increase ecosystem processes. Or alternatively, what you could have is that as you add more species within the system, you could have some species that are similar in terms of their functional trait. And so adding more species, particularly if you're adding species that have a similar functional trait, you might see a plateau in terms of its effect on ecosystem processes. So it may not be that just adding more numbers of species is going to see a bigger increase in ecosystem processes, particularly if you have redundant species or species that have similar ecological roles. So when we're talking about functional traits, we're talking about characteristics of individual organisms that impact their fitness through effects on growth, reproduction, and survival. And there can be a lot of different functional traits. We often define the functional trait based on the ecosystem process that we're most interested in. Now, functional types are a group of species that have ecologically similar functional traits with respect to their effects on ecosystems or through their response to environmental change. Just an example here is that there are several different species that form a symbiotic relationship with bacteria to fix atmospheric nitrogen. A functional trait is having that symbiotic relationship with bacteria to fix atmospheric nitrogen. A functional type would be something like nitrogen fixers. So all those plant species that have that symbiotic relationship with nitrogen fixing plants or bacteria, sorry. Excuse me. So I'm going to give you some examples, and we're going to first start about some examples of ecosystems within the Arctic of Alaska. So here are two examples of different types of graminoid-dominated tundra. One is called a non-acidic tundra, and the other is acidic. Excuse me, and that's based on the pH of the soil. In non-acidic and acidic tundra have several different species. They have several different grasses. They have some sedges. They also have some shrubs. All of this vegetation is very short. This is in the Arctic where we have long, cold winters and short summers. And the vegetation doesn't get very tall. However, we can have shrubs like willow and birch or betula, but they're just really small or short. In contrast, we can also have ecosystems in the Arctic called shrub tundra. And within the shrub tundra ecosystem, we can have different um, types of tundra that we could lump into their height and their dominance. So some ecosystems where we have shrubs, but they're kind of in equal proportion to other plant species, and they're short. We can have medium shrub tundra, and we can have very tall shrub tundra. All right, so here's just an example of some species found in moist acidic tundra. Excuse me. And I have them divided up by their growth form, which could also be considered a functional trait. So we've got a couple graminoids, and remember graminoids include grasses, sedges, and rushes. I have there's some deciduous shrubs. So these are shrubs, they're woody, they have woody stems, but their leaves they drop their leaves at the end of the growing season. And then we have some evergreen shrubs. So again, these have some woody material, branches, but their leaves they hold on to throughout the year. And as we talked about before, when we talked about litter decomposition, that deciduous versus evergreen can decompose differently, so they can have a different function within the system, specifically with things like decomposition or nutrient turnover. And then we have several different types of mosses. So in the Arctic, we can group our plants into different functional types. You could also do this in other ecosystems as well, but for an Arctic tundra plants, we can consider first their height. We have some tall plants, And if we have tall plants, they're usually trees in the Arctic, although we don't have that many trees in the Alaska Arctic. Or they could be short plants. They could then be vascular plants. So these are plants that have a true root system versus non-vascular plants. And within vascular plants, we can have woody plants like shrubs versus herbaceous plants like graminoids. Um, Within the woody, we can have deciduous shrubs or evergreen shrubs. And then in herbaceous, we can have, this is referring to different root types. So we can have sedges versus grasses or forbs. And then mosses, we can further divide into different functional types. You could do the same type of thing with another biome or another ecosystem. Your functional types or growth forms might look a little different. So when we identify plants by functional types, we look at groups of species that have similar functional traits, so like species that are nitrogen fixers or species that are deciduous versus evergreen. They tend to be related to phylogeny, growth form, and life history. Most useful, the functional type designation often tends to be most useful in low diversity systems like tundra, um, and most useful when response to and effects on multiple environmental factors and ecosystem processes are similar. It can also be most useful when defined in relation to specific ecosystem processes or functions. So if we're looking specifically at nutrient cycling or different trophic dynamics. So when we're looking at species identity, how does who's there affect ecosystem processes? So I'm gonna give you an example that's specifically looking at nitrogen cycle. So thinking about who's there in relation to nitrogen cycle, the plant species that are there that have that symbiotic relationship with bacteria that fix atmospheric nitrogen, that is one functional trait. So do we have an infixer within the system? We can have species that can take up nitrogen at different times. So we might have some species that take up nitrogen earlier in the growing season, while some species take up nitrogen later in the growing season. We have different plant species that can take up different forms of nitrogen. We talked about that most plants use the inorganic nitrogen form of ammonium and nitrate, but some plants can take up dissolved organic nitrogen or amino acids. The other we can look at is turnover time and allocation. So how quickly do these plants turn over their leaves? You know, if they're deciduous, they drop their leaves at the end of the growing season, which then that nitrogen in those leaves can become available for decomposition. However, if they're evergreen, they're going to hold on to their leaves for a couple years. So it's going to take a while before the nitrogen in those leaves can get decomposed and get incorporated in the soil. We can also think about litter quality. Do they have higher nitrogen content? Do they have other carbon compounds like cellulose or lignin that can influence their decomposition rates? And then we can also think about nitrogen loss. So here's an example of a introduced tree species in Hawaii, and it's a nitrogen fixer, Morella or Murica faya. So again, it has a symbiotic relationship with bacteria to fix atmospheric nitrogen. Now in this study, they looked at parts of the ecosystem or treatments where the Morella infixing tree was present versus absent. So they did a comparison with Morella and without Morella. And then they looked at 3 different ecosystem processes related to nitrogen cycling. So the first, they looked at nitrogen inputs, so how much nitrogen is getting input into the ecosystem. The second one, they looked at mineralization. And remember, mineralization is the conversion by bacteria of dissolved organic nitrogen to ammonium. And then they looked at the concentration of nitrogen in the litter. So if we look at first inputs, we see that when the system is dominated by Marilla, this infixer, that there's more nitrogen going into the system than when it's absent. We're also seeing higher rates of mineralization, so higher conversion of dissolved organic nitrogen into ammonium. And we're also seeing higher litter nitrogen concentration. So the influence of this one tree species can have a dramatic influence on the different ecosystem processes related to nitrogen cycling. So why would the presence of marilla lead to faster nitrogen mineralization? So or marilla increases both the inputs of nitrogen and the cycling rates. So if you've got higher litter nitrogen and you have more nitrogen going into the system, that higher litter nitrogen is going to decompose more quickly. And then there's more dissolved organic nitrogen in the ecosystem that can lead to more mineralization rates. So overall, we're seeing a so more nitrogen in the system and then faster cycling of that nitrogen, which then makes it more available for the plant. And just an example, in the first 15 years of succession on Alaska floodplains, alders, which are deciduous shrubs that also have a symbiotic relationship with nitrogen, alders have a symbiotic relationship with bacteria to fix nitrogen, contribute to about 75% of the nitrogen that accumulates over 200 years. So these nitrogen fixers within the system can have a pretty big impact on ecosystem processes and also in ecosystem development. So we're going to talk a little bit too about like root diversity. So who's there in terms of plants? So not only above ground, what is their different functional traits, but plants can have different functional traits below ground. And here's just an example of the diversity in the mass of roots and then the root length within the soil across different plants. So when we're thinking about what's going on below ground, plants can vary in their amount of the forms of nitrogen that they can take up. So here's a bunch of different plant types, and then we've got two columns. One is the ammonium to nitrate ratio, and the other one is glycine, which is amino acid, to ammonium ratio. If the ratio is greater than 1, then that indicates that first form of nitrogen is absorbed preferentially or more over the second one. So if we look at this first one, Arctic vascular plants have a 1.1 ammonium to nitrate ratio. So that suggests that they are almost equally, maybe a little bit more taking up ammonium versus nitrate. But they have a 2-to-1 for glycine and ammonium. So that means for every 1 unit of ammonium, they're taking up 2 units of glycine. If we look at our non-vascular plants in the Arctic, their glycine to ammonium ratio is at 5-to-1. So they're taking up a lot more glycine than ammonium. And we can kind of look down here on boreal trees. Boreal trees, which we can also often think of like spruces, take up about 19 units of ammonium per 1 unit of nitrate. So just kind of shows that different plants can take up different forms preferentially. So that's can be a functional trait, right? We've got different plants tapping into different resources of nitrogen. And some of that is probably due to reducing competition. So if they're all competing for ammonium, there's going to be some winners and losers. So we might have some plants that have adapted to take up other forms of nitrogen. And here's just a graph looking at, on the x-axis, we have the delta 15N of the litter or tissue of the plant. And I'm not going to go into a lot of detail, but 15N is a stable isotope of nitrogen, and it's in the atmosphere found in very, very low concentrations. So people often use 15N, they inject it into the ecosystem, like into the soil, so that it can trace where nitrogen goes, because the amount of 15N in the background or in the atmosphere is much lower. So that's so they're looking at kind of tracing nitrogen. A 15 delta 15N or a negative 15N means greater nitrate uptake versus a higher 15N means greater ammonium. And so we can look at different growth forms of different plants and see that they vary in terms of which type of nitrogen they take up. Also, plant functional types can have an influence on leaf litter decomposition rate. So here's just on the x-axis is a bunch of different plant species grouped by those plant species that have deciduous leaves versus those who have evergreen leaves. And then on the y-axis, we have mass loss from the litter. And so we see that we get more mass loss during the process of decomposition with those plants that have deciduous leaves versus evergreen leaves. And if we think about if we know the chemistry of these leaves, deciduous leaves tend to have more nitrogen in them. Evergreen leaves tend to have less nitrogen and more carbon structural compounds like cellulose, maybe even lignin, and maybe other secondary compounds that make them harder to break down by microbes. So plant litter traits can reinforce site nutrient availability, where if we see a decrease in growth of a certain plant, so maybe we see a decrease in growth of a nitrogen fixer, that we could then see a decrease in the litter quality, which could then influence, if you have poor litter quality, there's less nutrient mineralized by the microbes, and then that just leads to less nitrogen available for the plants and then less growth. In contrast, if we can see that plants that tend to have higher like litter quality, say like a nitrogen fixer, if we have more of that growth of that nitrogen fixer, we have more inputs of greater quality litter, which then leads to more nutrient mineralization, more nitrogen available for plant growth, which then leads to a positive feedback for more growth. Remember, growth is often limited by nitrogen, and so if we can increase the rate of cycling and the amount of nitrogen that becomes available, that can lead to acceleration or a positive feedback to more plants that can increase nutrient cycling and nutrient availability. So this is just looking at a bunch of different species that were planted in a monoculture of 5 different perennial grasses. And then it looked at the nitrogen mineralization rates in the soil across these different perennial species. So these perennial species also varied in their carbon to nitrogen ratios, so their litter quality. A high carbon to nitrogen ratio, like this first one, this has 44. That means for every unit, 1 unit of nitrogen, we have 44 units of carbon. So we call that a high carbon to nitrogen ratio. So that's going to be less yummy or less poor quality litter than well, I guess across all these, that's actually a lower quality compared to the 120, the CNN ratio of 122. So basically, plant species that have a lower, a lower seed to end ratio, which means higher litter quality, we see higher rates of mineralization. If that plant litter is yummier and easier for microbes to break down, they're going to break it down and turn over that nutrient. So when we're thinking about species richness, we may have a question of how do species number affect ecosystem processes? If we have, say, 1 species within the system versus 15 or versus 20, do we see an effect on ecosystem processes, such as like nutrient cycling. So is there an effect of diversity per se that's independent of identity? So this is just looking at the number of species and not so much looking at their function. So just looking at if we increase the number of species regardless of their function, do we see an effect on ecosystem processes? And this question has been studied pretty extensively in a series of experiments at the University of Minnesota Biological Field Station. So this was like an agricultural field which they cleared, and then they put treatment plots that looked at different combinations of the number of species. So each of those little squares you see in the photo, they have different number of species treatments, and then they looked at the effect on different ecosystem processes. And this is representing, and the type of plants they have in there represent a perennial prairie ecosystem, and this is in the Midwest in Minnesota. So there's been a lot of studies coming out of this, but one kind of early on study was specifically just looking at the number of species and then different different ecosystem processes. So if we look at that bottom graph first, we have species richness treatments, starting from, I think the first one's like 1 or 2 species, all the way up to 25 species in one treatment plot. And then they looked at nitrate in the rooting zone, so the amount of nitrate available in the rooting zone. And what they saw is that you see actually a decline in the amount of nitrate available in the rooting zone when you have higher species richness. The top graph is looking at species diversity, so it's just the number of different species. They've got multiple treatments. And then it's looking at plant biomass. So in contrast to what we saw in nitrate in the rooting zone, is that as you increase plant species diversity, you start to see an increase in plant biomass. But that starts to plateau. It sort of seems like once you reach 5 or like 8 species, the effect on biomass isn't more. So, there seems to be a limit on the number of species and its effect on plant biomass. And you might be thinking about, like, why is the nitrate in the rooting zone lower with greater species diversity? It's most likely that you have more plant species, you have more plants taking up nitrate in the soil, so then you would have less nitrate in the soil. Okay, so what are sort of the mechanisms? So they kind of came up with kind of 2 explanations for what the patterns they saw. Is that one is this complementary resource use. So as you increase species, more complete resource use. So you've got lots of different species and they're all tapping into potentially different niches or different nitrogen forms. The other effect is called the sampling effect. So as you increase the number of species, you increase the probability of getting a species with a strong effect on processes. So just with more species, you're more likely to get a few species that have a really strong effect, say, on plant biomass or nitrogen mineralization. So there was another additional studies kind of trying to partition out when we might see a complementary resource use versus a sampling effect. So in another experiment, they looked at growing species in monocultures versus mixtures. So the idea is that if they are expecting to see a complementary resource use, that the production in a mixture of species would be greater than the production in the monoculture. However, if you expect to see a sampling effect, the production in the mixture should equal the production in the monoculture. And there at the time, there was very few well-controlled studies that tried to tease these apart. So this was one of many of what we call Tillman's experiments. So Tillman was one of the scientists that kind of helped start this whole series of like research studies looking at biodiversity effects on ecosystem processes. He helped start the whole LTR Minnesota prairie site. And so the main goal for this particular study that they published in 97 is that experimentally species richness and so they wanted to experimentally separate out the species richness effect versus the composition or functional diversity effect. So they had two treatments. The first one was looking at just the number of different species. So they had, a control where they had no species, and then treatments where they just had one species, and then treatments with 2, 3, 8, 16, and 32 species within a plot. They then had a second set of treatments that specifically looked at functional groups. So they had no functional groups represented, only one functional group, 2, 3, 4, and 5 functional groups. And here's some of what they found. The top graph is looking at just the number of different species added, and then plant biomass. And as you see, you can see that as you increase species diversity, they also saw an increase in plant biomass up to a point. And then the bottom is looking at functional diversity in plant biomass. And you see kind of a similar pattern where at some point it starts to plateau. So kind of the results from this is that the functional diversity explained the greatest amount of variation in plant biomass. And when functional diversity was included, species diversity actually had no effect on its own. So they suggested that because we saw the same response when you added just the different functional groups and not necessarily individual species, that they concluded that the functional diversity had a stronger effect than just increasing number of species. So it doesn't necessarily matter how many species you have, but how many functional groups are being represented within that ecosystem or that treatment. So here's another where they looked at functional diversity. So we've got one functional group, two functional groups, and three functional groups represented. And the functional groups they're looking at here are whether it's a legume, and legumes are nitrogen fixers, so they have that symbiotic relationship with bacteria to fix atmospheric nitrogen. They're looking at C4 grasses, so C4 grasses have a photosynthetic pathway that helps them grow in low water and high light conditions. And then they looked at the combination of C4 grasses and legumes together. So the first one is looking at, the first functional diversity is looking at they've got legumes by themselves, C4 grasses by themselves. The second is looking at legumes and C4 grasses plus C4 grasses and legumes. Or sorry, yeah. And then the third is also looking at that combination. So what they found is that um, is that they saw greater influence. So if we think about the height of those bars, when C4 grasses and legumes were combined, than when they were planted separately. So the effects of functional diversity were caused by the presence or absence of key functional groups. And over-yielding was also observed where C4 grasses yielded more in higher than in lower diversity treatments. So in conclusion, the effects of functional diversity on ecosystem processes were caused by the presence or absence of key functional groups. Diversity per se, so just the number of species, only had an effect on ecosystem processes via the presence or absence of certain key species. So there's a lot we don't know, or there's also a lot that has been studied since then, but we don't have enough time really in this class to dive into each of those separately. But since these studies, there's been questions and studies thinking about. So we are looking specifically here at the plant species diversity, but what about microbial diversity? How does that play a role? Again, is the number of microbes more or less important than the function of those microbes? So what role does microbial diversity play in ecosystem function? And is function impacted by microbial diversity? So those are some other questions that came out. Some other unknowns is, where do natural ecosystems fall on this curve? So the Tillman data is from an experimental design where they created these treatments. But if we go out into nature and we observe different ecosystems and their functional and species diversity, do we see the same pattern? And how important is richness in explaining variation in production in natural ecosystems? So richness may be most important in low diversity systems, either managed or human impacted systems or natural systems. And how do effects change over time? So now we're going to look at some results from a paper that looked at where natural ecosystems fall on this curve. So this is a paper that was published in Nature back in 2017, and it's essentially a meta-analysis, so looking at multiple studies published. And this meta-analysis looked at 67 field studies, 133 estimates. It focused on natural versus managed ecosystems. It looked at also empirical studies, and then it looked at diversity gradients to look at the impact on biodiversity on ecosystem function. And then they did a further statistical approach to try to separate out diversity versus different environmental covariates. So here's we're going to just look at a few things, a few highlights from the study. So here's looking at the log response ratio. So when you do a meta-analysis, it's a study looking at all the published data on that and looking at the effect sizes. So each of those research papers measured some treatment effect, and then you can plot all those treatment effects from all those different papers and then look at patterns in terms of its effect size. So here they use one statistical analysis called a log response ratio to measure the effect size. So if the value is over 0, that means it had a significant positive effect, that study did. So the red is different experiments from the different studies, and the blue represents observational studies. And the number next to that is the number of studies. And the first part here is the first row here is looking at the effects of algal species richness on algal biomass. So we saw that there was over 1,000 different observational studies looking at effect on algal species richness on algal biomass. The next one, they looked at the effect of plant species richness on grassland productivity. So over 1,000 studies there. And then they also looked at the effect of tree species richness on forest productivity, and then also the effect of herbivore species richness on algal epiphytes of eelgrass. So looking at different effects of species richness on different ecosystem processes. And you can see that all these studies had a positive effect of species richness on that ecosystem process. And then here's just some more of their data. This is looking at the proportion of estimates And so out of the different studies, they ranked, they looked at which studies ranked was the effect of biodiversity over other variables like climate predictors or nutrient predictors on biomass production. And so this first graph here is looked at the number of studies where biodiversity was ranked 1, 2, or 3 versus comparing it with climate predictors. So it ranked, 33 of the studies ranked biodiversity as the primary, with 30 studies ranking it as the secondary. And then the other graph is looking at the rank of biodiversity versus nutrient predictors. And biodiversity, 14 of the studies rank biodiversity as number 1 and 8 of the studies number 2. So in addition, we might ask, like, how does understanding biodiversity and ecosystem processes help us understand the human effects? Um, so, you know, does biodiversity help to buffer from different human drivers or human-caused environmental change? So this is another series of studies that looked at biodiversity loss versus other human-caused drivers of environmental change. This again was done out at Cedar Creek Ecosystem Science Reserve in Minnesota. They had 11 experiments. They looked at the mean length of this experiment. So they had some experiments that went over through 13. The average length of the experiments was 13 years. However, the experiment's time ranged from just 1 year to 28 years. And then each experiment manipulated one or more environmental change variable. So either nitrogen addition, watering, so changes in like drought or over precipitation, elevated CO2, fire, and herbivory effects. So here's some of the data they found, and there's a lot to go over. We're just going to look at graph A and B first. On the x-axis, we have the different experimental treatments. So we have those blue bars are looking at differences in the number of species. So we had some treatments that range from 1 to 16 species, some that range from 2 to 16, and some 4 to 16. We had different nitrogen addition treatments with 3 different concentrations. We had some studies that had a watering treatment, while some studies had a drought treatment. We had some studies that had a CO2 enrichment treatment, one studies that mimicked herbivory, and then some studies that mimicked fire. So graph A there at the top is looking at the biomass difference relative to the control, and we see the highest biomass difference were related to changes in species, and then secondary to that is nutrient additions. The bottom B graph there is looking at the log response ratio. So this is the magnitude of the difference, and we see a much larger magnitude when we're altering species richness and nutrients. And then that graph on the right is just like pulling all those together and looking for patterns where we see if we pooled all of the response ratios for species, we see that early studies early on, like the first couple years, that nitrogen addition showed a bigger response in biomass difference than species. But later in longer term studies, we see that the species or biodiversity treatments had a larger effect on biomass difference than the nutrient addition. So another question to think about is, like, in terms of human influences, is looking at crop diversification, biodiversity, and crop yield. So there's been some questions of, like, if we increase biodiversity in our croplands, will that lead to increased ecosystem services, but also maintaining crop yields? So this paper we're going to review here was published in Science in 2020, and it's looking at the diversification of different management practices versus simplified cropping systems. And again, this was a review of meta-analyses. So they looked at a whole bunch of different meta-analysis, and remember that meta-analyses are essentially looking at all the published studies and looking for patterns across those published studies. So they reviewed 98 meta-analyses, and then they looked at, they did what they called a second order meta-analysis. So they did their own meta-analysis with the meta-analysis studies, and they had about 5,000 original studies with over 41,000 or almost 42,000 comparisons. Okay, so now we're going to look at some of the results. So let's first look at the first figure, figure A, there on the left. We've got different agricultural diversification practices, or ecosystem services on the x-axis, things like soil fertility, crop yield, nutrient cycling, carbon sequestration, climate regulation, water regulation, pest control, biodiversity, and pollination. So it's different ecosystem services. And then the y-axis is the effect size of those different treatments, with the green part of the bar being a positive effect, the gray a neutral effect, and then the red a negative effect. So if we look across that, we see a lot more green than we do red and gray. So overall, we are seeing like a general positive response. However, in some instances, like in crop yield and climate regulation, we are seeing some studies showing a negative effect. We now can look at that a little bit more closely and separate the different effects based on different land management practices. So on those top two graphs, we can look at organic amendment effects on those different ecosystem processes versus reduced tillage versus crop diversification or non-crop diversification, and then inoculation and organic farming. So we can fine tune it and look at different practices. Kind of a quick look suggests that adding organic amendments and reducing tillage tends to have more of a positive response on those ecosystem services, with a few negative ones. And then crop diversification seems to have a larger effect, larger positive effect than non-crop diversification. All right, so here's just kind of looking at all the effect size for those different ecosystem processes. The first graph is looking at all diversification practices put together, and we see that biodiversity and pollination tend to have a larger effect size than some of those other, they tend to have a larger effect size on those ecosystem processes than, say, like crop yield or climate regulation. And then if we look at above ground, the biodiversity, and it looks like water regulation tended to have a stronger effect, or we tended to see a stronger effect on biodiversity and water regulation for above ground. And then below ground, diversity is also showing having a positive effect on diversity.